My work involves the design and synthesis of inhibitors of human pancreatic alpha amylase as a means of controlling blood sugar levels in diabetic and pre-diabetic individuals. In unaffected individuals, the consumption of starch and the subsequent activity of alpha amylase leads to a small but relatively inconsequential peak in blood sugar levels. However, in individuals with diabetes, the activity of alpha amylase leads to a large and prolonged increase in blood sugar levels, which can have a number of harmful side effects, especially in the long term. So as such, we aim to regulate the activity of human pancreatic alpha amylase as a means of preventing this harmful increase in blood sugar levels and improving the life of those with diabetes. Alpha amylases are not unique to humans. Many organisms, such as many insects and bacteria, also produce alpha amylases as a means of breaking down starches in the plants that they feed on. And as a result, many plants have produced alpha amylase inhibitors as a means of defending against these predators. We wanted to tap into this natural resource of alpha amylase inhibitors in hopes of finding a new compound that could be developed into a therapeutic. Our top hit was extract of the bulb of the common garden plant, Crocosmia crocosmiflora. We isolated the active ingredient from the bulb, carbohydrate derivative called monbrendan. So monbrendan is a very specific and potent inhibitor of human pancreatic alpha amylase. Unfortunately, monbrendan is only produced in small quantities in the plant, and its complex structure makes it difficult to synthesize. So one of my projects at UBC is basically to design and synthesize simpler analogs of monbrendan in hopes of finding a molecule that has the same potency and specificity while being synthetically accessible.
My main project is on Gaucher disease. Gaucher disease is a genetic disease or a lysosomal storage disease. It's caused by a mutation in the gene that codes for a particular enzyme, which is called glucocerebrosidase, that is responsible for the breakdown of fatty glycolipids coming from worn out and dying cells. When this enzyme is deficient, that fatty molecule accumulates in a cellular organelle called lysosomes, and the concentration builds up and eventually it becomes toxic, provoking a series of symptoms that are that can range from mild to deadly. Currently, the most common therapy for patients of the Gaucher disease stands on administration of recombinant enzyme to replace the enzyme that they are not able to produce properly. In my case, my approach is to design inhibitors of that enzyme that help stabilize the proper folding of the enzyme, and then so that more of the enzyme mutant that is produced in the body gets trafficked into the lysosome, where it has to degrade all the accumulated material. So the advantage, or the main advantage of my approach is that we don't need to infuse patients with exogenous proteins. We are trying to fix somehow the protein that they generate in their cells that somehow doesn't get transported into the lysosome where it has to work.